So translation, so whatever genetic information is there on the DNA, it will be like red, and it will be coming into the cytoplasm from the nucleus in the form of RNA, isn't it? So that's what we have done with transcription. That means now messenger RNA, rRNA, and tRNA are ready. They are all synthesized by the mechanism of transcription. So once the transcription is done, that means RNAs are ready. We know like function of RNA is to synthesize proteins, right? Biosynthesis of protein or polypeptide. So whatever
Yes, ma'am.
information the mRNA has brought, based on that information, you will see tRNA will be bringing the amino acids, and the chain length of the polypeptide chain will keep growing till the stop signals come, and you will see there will be complete formation of proteins. So that's what we are going to learn when we do now this topic, translation. Okay. So the genetic information stored in DNA is passed to RNA through transcription, right? And ultimately, synthesis of proteins is taking place by the mechanism of translation. Okay. Now, this is important short note beta, genetic code. What do you mean by genetic code? See, like when you have seen like Tata box, Hogness box, these are the terms we have come across when we were doing transcription. That means you have seen a sequence of genetic code, like it can be AGC, anything, adenine, guanine, like cytosine and uracil. So these are the sequence of genetic information, like 333 information of this codon, that is actually called as code word, and each code word will be coding for one amino acid. Okay, the three nucleotide base sequences in mRNA that acts as a code word for amino acid. So here you have few examples better, like UUU. It's a genetic code which is coding for phenylalanine. UUC is also coding for phenylalanine. Like that, we have so many genetic codes which are coding for different amino acids. Okay, leucine, lysine, proline, threonine, serine, arginine, glycine, and so on. And you see there are certain stop signals also, like which are given in red color, like in the whole chain of mRNA, if the tRNA comes across UAA or UAG or UGA, that means they are not coding for any amino acid, and the chain length of polypeptide chain will stop. So these are called as stop codons or stop signals. Okay, and there are certain characteristic features of this genetic code. See, these codes or code words are actually very much like universal. That means whether is in plants or human beings or any living organisms, they are going to code for same amino acid. That is the meaning of universality. UUC, they always code for phenylalanine. Like that, you have UCU and so many other four different code words. They will always code for serine. Okay, like that. Yeah, that means they are very much specific. Okay, so we know like these three codons, they are not coding for any amino acid, since they are called as termination codons. Okay,
Yes, ma'am.
and then the characteristic features of this genetic code are universality. As I said, they are always coding for same amino acid irrespective of any living organism. Specificity, a particular codon always codes for same amino acid. Hence, genetic code is highly specific, non-overlapping. That means, see here, they are written continuously. There is no overlapping. U is not lying above you or below you. They are written in continuous form. Okay, and there are no hyphens or there are no obliques or anything should not intervene while you are writing the sequence of this genetic codon. Okay, that is. And we have seen frame shift mutations, no, like a particular frame will be cut and it will be attached to either the extreme one end of the whole of the genetic information. So those mutations are very dangerous, because They will be coding for different amino acids which is not existing, right?
Yes,
There
ma'am.
is a degenerate code. What that means? What actually, if you see, we have only 20 amino acids, but if you just check here in this table, we have different, so many different, as many as 60 code words are there. That means what? 61 codons are there, but most of the codons are coding for one same amino acid. For example, if you see here, these are four different code words, but all four are coding for serine only.
Again.
These are
Yes,
five
ma'am.
different code words, around four again. That
But
is
for proline.
proline. That means what? Degenerate. That means more than one code word can code for only one type of amino acids. Okay. So that's what is actually called as Google hypothesis. One more thing we should learn in Google hypothesis is, if you just observe these multiple code words, first two nucleic acids are same, UC, 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 CU, 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 AC, 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 but only the last one is different, isn't it? UCAG is the last code word you are finding. That means what? The last code word is generally overread by tRNA. Only the first two are taken as standard, based on which only the amino acid will be brought to the site of protein synthesis. That's what is Google hypothesis. Okay, got it, beta?
Yes, ma'am.
Now just look at this picture. This is mRNA. This has so many codons: AUG, GUG, CUG, CUA, something. Each code word is coding for one amino acid. Now, this is mRNA which has brought information from the nucleus into the cytoplasm. And this is tRNA. We have around different types of tRNAs. As many code words are existing, so many tRNAs are existing. Okay, that means around 60 tRNAs we have. So, like suppose on the mRNA, if you are finding the code word AUG, which is genetic code, so correspondingly, the anticodon area will have, we know like A is base pairing. Do you remember the base pair ATGC? A always
Yes,
paired
ma'am.
with T. G, there we have triple bond, G pairs with C, double bond, right? So, but in case of like RNA, instead of T, we have U, right? Thymine is replaced by uracil. Hence, you are finding instead of ATG, you are finding AUG. So, adenine pairs with uracil, AU, UA, GC is same. So, in like the codon, if it is AUG, anticodon would be UA, C beta. Okay, so and this is coding for which amino acid? Methionine. So once you see the structure of tRNA, you will see it has an anticodon area. It has complementary binding area. Okay, and this area, like it picks the amino acid like methionine. Okay, and it brings to the site of protein synthesis. So anticodon, codon, 1 minute. So, like mutations, we have already studied, like frame shift mutations, you have an idea. So, that is all about genetic code beta. Once you know the genetic code, then we will talk about protein biosynthesis. So, this, in case if the examiner wants to give you tough question as essay, even protein biosynthesis can come as an essay question beta. So, it has certain steps like requirement of the components, activation of amino acid, then protein synthesis proper, and chaperones and protein folding and post-translational modifications. Now let us start with the requirement of components. Obviously, for the synthesis of proteins, what do we require? We require amino acids. How many amino acids are there? 20 amino acids, of which 10 are essential, like we study in chemistry part. AV
Okay.
Hill MPTT is the code to remember the essential amino acids. Then we know ribosomes are the factories where protein synthesis is taking place. Now, this is a very good picture which gives you a complete concept of what is happening during protein synthesis.
Protein biosynthesis.
We know like we have three different types of RNAs like ribosomal RNA, messenger RNA,
Messenger transfer
transfer RNA,
transfers RNA.
mRNA, RNA. Very good. If you just see this structure, this is actually ribosome. Okay. So in this ribosome, you are finding like there will be a small unit and a large unit. So ribosomes are carrying the RNA. What RNA is it carrying? mRNA. So this is ribosomal RNA which is given as an outline of this round picture, and this wavy line is indicating messenger RNA, and tRNA will be bringing the amino acid and it will be increasing the polypeptide chain length till it gets the code for stop signals, termination codon. So three RNAs are actively working here. This is ribosomal RNA. Okay, here inside the ribosomal RNA, messenger RNA has brought the information like AUG, GUG, CUG. Okay, like different types of code words are here, and based on these codons, tRNA is bringing the amino acid and it is attaching like this. Suppose one amino acid, beginning of protein synthesis, methionine has come in response to AUG. Again, CUG is there, then phenylalanine has come, CAA is there, leucine has come, then lysine has come, arginine, methionine. So many different types of amino acids have come based on the genetic code which is available. So UAA, there are certain stop signals, right? Once that stop signal has come, you will see they are not coding for any amino acid. Hence, this polypeptide chain will not grow in size anymore, and whole of the protein synthesis has occurred. And whatever like a polypeptide chain which you are seeing here, you call it as polysome. And this complex series of ribosomes where we have ribosomal RNA, messenger RNA, and transverse RNA actively working, you call this complex as polyribosome, okay, which are working during protein synthesis. Everything you can do with the help of diagrams. The same thing, if you just read once, you will get an idea like what is happening. Now, protein synthesis proper. See, amino acids. Suppose if it, like we have just talked about methionine, no? So let's keep methionine only. So methionine, if it has to go and get attached to the tRNA, this amino acid is actually taking the attachment of AMP so that it is activated. E is energy. So here ATP is releasing the high energy phosphate, that is PPI, in the presence of amino acyl RNA synthetase. So this activated amino acid, that is AMP amino acid, now it is attached to the tRNA. It is loaded now so that it can go and form a chain of polypeptide. Okay. So once it is going and getting attached to the T terminus of this transfer RNA, you will see energy is released, AMP is released. Okay, that is. So that is the formation of, you call this as amino acyl tRNA. Okay, you draw
Yes,
this
ma'am.
and give your own explanation in your own words. So initiation of translation, there are four steps, beta. Ribosomal dissociation. Basically, in the ribosomes, we have two Svedberg's units. No, like we have one half, the other half. Overall, these are 80S, which is now split as 40 and 60S. Okay, so formation of 43S subunit, proteination complex, formation of 48S initiation complex, ATS initiation complex. These are the four steps which are occurring. So what you have to do, Shubhangi, see, better you learn these pictures better, whatever is given, a diagrammatic representation of initiation of protein biosynthesis in eukaryotic cell. Okay.
Yes, ma'am.
So few symbols are given like one square, one triangle and circle. Okay. So just learn this better. It looks little complicated, but don't keep trying to understand everything. Just learn this. Okay. Like these ribosomes are
Yes.
just being split. Okay. So the mechanism is you are finding finally what is this red shown? tRNA is shown, right? So tRNA is
Yes.
bringing methionine only, what we have kept as standard. AUG coding for methionine, no? So this is indicating mRNA, and this inverted you are finding a structure like a vessel. So this is ribosomal RNA, and red in color is transfer RNA. Okay. So just because you have time now, it's better you learn this better, because all other things which are easy, you already know so much. But things which are rarer ones also we will not leave. So please learn this better.
Yes, ma'am.
So అది రాస్తే మొత్తం ఆన్సరే అయిపోయినట్టు, like one point if you just read you will get some idea, but looking at that flowchart only examiner will give you full marks without any doubt. Okay.
Yes, ma'am.
So termination of translation. We already know termination. What are the stop signals, beta? UAA, UAG,
You,
UCA, no?
you see. Yes, ma'am.
Inhibitors of protein synthesis. So we had inhibitors for replication. We had
For
inhibitors for
transcription.
transcription. Now even for translation, we have certain drugs like antibiotics, okay, which can stop the process of protein biosynthesis. Okay.
Yes, ma'am.
Even this also, this is much easier than the previous one, but what it shows is you are finding the outline of ribosomal RNA, you are finding an outline of messenger RNA, you are finding also an outline of this tRNA, isn't it? So now you see one tRNA, suppose if it is coding for methionine, one more tRNA is coming and it is coding for second amino acid. He didn't mention the second name, like just amino acid 2 is written, AA2. Like that, you will see one more tRNA will come and it will be coding for one more amino acid. So as many RNAs are coming, you will see so many amino acids are getting added to polypeptide chain. Isn't it better? And
Yes,
your
ma'am.
ribosomes are finally split into 60S and 40S, and the protein chain is released. And even the mRNA is coming out, which was initially inside the ribosome. Once its
Ribosome.
job is done, the Swedberg units 40S and 60S, they are also like getting separated. Okay. So like that, see, so many diagrams are there. Once you just draw and write the explanation in your own words, it itself will make your essay answer if it comes like almost 6 pages, like 3 pages, 6 sides, right? Like that it becomes. That is more than enough. घुमा फिरा के इवन ये भी वही है बेटा. These are all tRNAs only. Amino acid 1, amino acid 2 and all. Huh? This is peptide bond formation which is shown. You can just also give this how the peptide bond is formed. So for the see, this is amino acid 1 beta, like suppose this is methionine, suppose this is lysine. So in between them you are finding there is one peptide bond formed like this, but you need not remember this structure, absolutely not required. You can just write amino acid 1, amino acid 2 and just peptide bond. You can just give the binding point over here. Just show this enzyme peptidyl transferase working with the loss of one water molecule. Okay, that helps in the formation of peptide bond. Okay.
Yes, ma'am.
Then we have post-translational modification of proteins. So we have done like post-transcriptional modifications also. Whatever RNA comes out of the nucleus, I said it is in very much crude form. Hai na? So it
Yes,
will
ma'am.
have so many changes. Methyl capping will be done. SNRPs we have seen. Okay, it has certain areas which are not coding for anything. So many changes we have seen. Similarly, even the protein which is formed, the polypeptide chain which is coming out after the extensive work by these three RNAs, it will also have certain changes. The polypeptide chain, what does it undergo? Even this itself may come as either 3 marks or 5 marks, but more than translation. Frequent question is this only. Post
Okay.
translational modifications of proteins is the most frequent question. Okay.
Yes, ma'am.
Huh. So you are finding once that polypeptide chain comes out, which is in crude form, so it will undergo certain changes, like it will start folding on itself. You call that as tertiary structure. There may be proteolytic cleavage, that means proteins like pieces of polypeptide, okay, intense splicing, like we had in the previous thing also, like exons and introns splicing. So the same mechanism is also seen here. Then covalent bonds, okay, new chemical groups are added. So just read this in detail also, so that will help you because it's very important. Okay.
Yes, ma'am.
Like he gave like even post-translational modification through their amino acids. So amino terminal amino acid post-translational modification would be glycosylation. So many, at least one. See, give this table, but remembering all 20 amino acids, 10 and all becomes difficult. Choose any 5 which are easy for you, like arginine. Post-translational modification is it undergoes methylation, proline hydroxylation, methionine sulfoxide formation, like that. Lysine acetylation, easy things better. Cysteine, cystine formation, like that. 5 easy ones you pick and just give the table so that your answer also looks complete. Okay.
Yes, ma'am.
Just checking if anything is left over because this is the most important topic. You have to give details of everything. Just read once,
Hmm.
you definitely understand. Protein targeting is not required. Mitochondrial DNA also same thing will happen, but it takes place inside the mitochondria. Transcription and translation. Suddenly, if that question also comes, you give the same process. Nothing to worry.
Yes, ma'am.
So that finishes the chapter.